Okay, so I start off by welcoming you, Dr. Yogesh. Your topic for the day is Genome and Ploidy, understanding the overall need for pre-implantation genetic testing. So, Dr. Yogesh, he's a professor, academic director, and head at SEART, which is the School of Embryology Assisted Reproductive Technology, Sridhar University. He is also an ASHRAE IFS certified senior embryologist. He's a member of the Global Embryology Forum, and he is very inclusive part of our iSeat Academy. Very involved faculty with us, and he has done a lot of publications, and he is extremely interested in immunomolecular biology of IVF and assisted reproduction in male subfertility, and a whole lot of other things to his name. So welcome, sir, and let's get started.
Thank you, Dr. Meenu madam, for kind introduction, and special thanks to Talwar sir for considering me for discussing or telling something about this gene and genetics. So my presentation there, I want to share my screen. Mohit, please allow me to share it my screen, please.
Yes, sir, you can share.
Okay. Just a minute. I'm not able to. Is that no? I'm not able to share. With mine, yes. Sure. Yes. So basically, what exactly we are going on? Last speaker, Dr. Nisha madam. She spoke a lot about the big thing. But where these big things are coming? So as a as an embryologist, we need to understand, and as a service provider for the couples who are seeking this IVF services and IVF services at any of the IVF center where me, like an embryologist, or you, or someone like clinicians, and the one more person, genetist, who are sitting there to offer them in terms of their normal babies. And when it's talking about the normal babies, we need to understand that how can we counsel the patient and how can we understand the script of whole genetic testing. So we need to understand the basics of the genome and what kind of information we are getting. When we are seeing that aneuploid is more, aneuploid is that, abnormality is that, more abnormal embryos. So we need to understand from the basics that how these abnormalities in the embryos are coming from. Whenever we are having one infertile couple, we need to offer them, we need to treat them for IUI or IVF, and once it comes to the IVF, then you will have to have the embryos, and your duty and my duty is to have a good quality embryos, which is euploid. So what is the currently status of this genetic testing overall? So we will discuss everything slowly, gradually through the slides. And here are some slides you need to understand. First, the genome and the ploidy, and what is the need? What is the need of pre-implantation genetic testing? We need to understand everything all together first, so that we can tell them something, that what is happening at the level of the genetics of the embryos, and later on their prospective children. So ploidy in genetics is the number of the chromosome occurring in the nucleus of a cell. So whenever we are talking about this ploidy, nucleus are having specific number of the sets of the chromosomes which should be normal. Okay. And whereas when we are talking about the genome is the entire set of the DNA instructions found in the cells. In human, the genome consists of 23 pairs of the chromosome located in the cell nucleus, as well as small chromosomes in the cells mitochondria also, because there are some of the areas where mitochondrial diseases are also considering to the offspring. In humans, we are having somatic cells, and somatic cells means we are having 23 pairs. So we are diploid in nature, and during meiosis, the cell produces gametes, and these gametes are sperms and the oocytes, and then these two copies are divided into are reduced into half, and then the condition is called as haploid. So we as a whole are diploid, and the gametes which we are producing, they are haploid in nature. And these two germ cells and the the sperms and the oocytes, they unite together, and then they again restore the condition of the diploid when babies are formed. So we need to understand that how this haploidy and diploidy is just reducing and restoring. Genome means where we are considering one cell, and cell is having nucleus as well as the mitochondria, and then this whole nuclear genome plus a small nuclear chromosomes in mitochondria, they all together, and these genome contains all the information needed for an individual to develop and functional normally. So we need to understand first that what is the normal and what is the then we can understand the abnormality into the genome and genome of the embryos later on. So how these are? There are nearly 3 billions of the nucleotides, and to date, only humans are the only species. Those are successfully sequenced to its own genome. This is the reason we are talking about whole genome sequencing, aberrations, microdeletions, trisomy, monosomies. This is the only reason that we have the Human Genome Project, where complete genome, all the sets of the DNA sequence has been identified to a reason that we can identify specific genes of a specific segment of the DNA. So the full range of the RNA molecule which is expressed by the genome is called as transcriptome, where when any of the problem happening at the level of the gene sequence of the DNA, there is a whole change in the sequence of the RNA, and then there is a change in the RNA, then ultimately there is a whole change into the full assortment of the protein that is called as the proteome. All DNA molecules are meant for transcribing into a specific mRNA messenger molecules, and then these messenger molecules are coding for a specific protein. And whenever we are talking about the abnormality, as a normal male or female, I am producing everything normal. And if there is something abnormality, then all those genes are producing something abnormal, which is not good for the human being. So this is the model of the DNA, which have already been discussed by the previous speakers, the very respectful speakers. Here, just to give a brief, we have 23 pairs of the chromosomes, 22 are autosomes. These are homologous chromosomes. Then by their size, number, size, shape, and number 1, 2, 3, and 4, and like 22 till 22, and 23rd pair is the sex chromosome. So some of the genes are sex linked, some of the genes are autosomes. So the sex linked characteristics are controlled by the sex chromosomes, whereas the autosome control only the inheritance of the characteristics, not the abnormality at the level of the sex link. So here to yes. So before understanding this genome and ploidy, we need to go through the karyogram. Karyogram is nothing but the ideogram. Ideogram, which is also called as ideogram, where you can see that normal level of the chromosomes sitting like this in a homologous 1 and 1, because they are in the pair, and till 22 they are same, but in case of male and female they are different. So these are called as the sex chromosomes. Though a karyotype is the general appearance of the complete set of the chromosomes in the cells of a species. It's not only about the humans, but whenever we are talking, we can carry out any of the DNA material of any of the eukaryotic cell, and we can find out that how many numbers of the DNA and the chromosomes are there. So karyotyping is the process by which the karyotype is discerned by the determining the chromosome complement of an individual, including the number of chromosomes and also any abnormality. So these are the ways we can understand that what is happening. So basic thing is that if we are having, whenever we are talking about the genetic analysis or genetic counseling of the patient, so karyotyping is the first thing that we can identify something at the level of abnormality, if it is there or not in terms of the karyotype is showing the extra chromosomes or missing chromosomes or monosomy, trisomy, we can find out easily. So how to read one karyogram is like they look like this, but when we are going to read, either they should be 23 pair, but sometimes there is a missing, one of the pair is missing, one of the chromosome in one pair is missing, it is called as monosomy, and one of the pair is having extra chromosome, and then it is called as trisomy. These are the aneuploidies. Okay, so in case of trisomies there are 3 chromosomes, and in case of monosomes there is only 1 chromosome. So these are the ways we are going to understand the karyotype. So when we are talking about the ploidy in eukaryote, it's like when haploidy is the single copy, okay, n number copies of the chromosomes, which usually found in the sperms and the oocyte, which we have already discussed. In case of diploid, 2 times of the number of the chromosomes, 2n copies, which are found in the which as found in the haploid set, but they are double in number in somatic cell of the body, and humans are diploid, as we have already discussed. In case of polyploidy, when there is a multiple sets of the chromosome that found in the haploid cells, like wheat are the hexaploids. But when we are talking about the polyploidy in animals, because they have more number of the normal number of the sex chromosomes, they are usually infertile or sterile. So for the animals, we are using the term sterile. So they cannot produce babies. But whenever we are talking about the more to the IVF and the euploidy and aneuploidy. So euploidy means one cell is having exact number, normal number of the chromosomes into its nucleus. Okay, so it is the state of the cell or the organism having one set of the same set of chromosomes which are genetically normal. So possibly including this, which exclude the sex determined chromosomes. But most human cells have two each of 23 homologous monoploid chromosomes, and in total these are 46. In case of aneuploidy, the states where one or more individual chromosomes of a normal set are absent or present in more than their usual number of the copies. So aneuploidy, it is unlike euploidy. Aneuploidy karyotyping will not be a multiple of the haploid number. Rather, it is an example of including having an extra chromosome. Down syndrome is the example, and when we are having missing, it is a Turner syndrome. So in case of aneuploidy karyotyping are given names like suffix zomi, not ploidy. Whenever we are talking about the aneuploidy, we cannot use the term ploidy. We will use the term somi, trisomy or monosomy. So these are the big chart of showing n number of the chromosomal abnormalities, and with their classification as per the autosomal or X or Y linked or translocation aberrations, leukemia. So these are the abnormalities which are there, and they can be there into the they can be there into the embryos when we are considering the IVF. So what is the relation of the aneuploidy with the embryos? When we are stimulating the ovaries, so the dynamic of the matrix spindle having some sorts of the events at the level of the alignment of the chromosome during the oogenesis and after the fertilization as well. So meiotic and the mitotic phase both are provoking the more aneuploid embryos. So this is happening because of the non-disjunction of the chromatids, unbalanced disjunction, chromosome scattering, formation of the aneuploid oocyte sperms and the embryos. So in case of patients who are undergoing IVF, cells are more prone to have these abnormalities because of the, in case of oocytes, it is controlled over hyperstimulation. In case of men, because more the cells are abnormal, and other confounding effects, the external effects, the intrinsic and extrinsic effects are there. Sir is also going to tell you about this epigenetic effect, which is extrinsic and intrinsic, both induced by the extrinsic and intrinsic. So these are the factors we will have to consider first when we are talking about the providing them understanding the genetic services to these couples. So genome encompasses the entire genetic material of an organism, including DNA and the gene expression, which plays a vital role in determining various traits and characteristics, making them essential to understand its role in the pre-implantation genetic testing. So it was found by some of the workers that cleavage stage embryos are go as high as 80% of the abnormality level. While in case of when we are discussing going to assess the abnormality level at the level of blastocyst, the rates of the abnormality are a bit lower. So this has led to the valid assumption that deselection of the embryos carrying such abnormalities would have a beneficial effect over the outcome of the IVF XCT cycles. Why? Because if we are able to find out something at the level of the gene and genome, then we can easily find out deselect all the abnormal embryos and select the good one. So here, what is exactly happening at the level of, so there are PGTA has endured several reiteration both at the level of technology, but it requires embryo stage for the biopsy or something. But initially when we are discussing these genetic analysis, when blastomere blastocyst biopsy was not there at that time, 8-cell stage, one single blastomere, two blastomeres were biopsied, and they were utilized. They were utilized by the comprehensive genome screening or using array comprehensive genomic hybridization for assessing the abnormality. But later, whole genome sequencing for the blastocyst biopsies is currently performing. So PGTA was initially proposed by the patients with advanced maternal age, since they are at the risk of producing embryos with meiotic abnormality. But several other patient categories such as repeated implantation failure, male fertility, and recurrent pregnancy losses are new targets now. So we can target as per the, it is not only about the advanced maternal age, but other aspects also. So why we are doing? So either we are doing PGT for aneuploidy or for monogenic disorders, like for understanding the mutations and the structural rearrangements. So these are the target specific genetic abnormality we can find out. So here it is the complete definition of the PGTA, how we are using it. So this PGTA is for this aneuploidy, M for the mutations, and SR for the structural rearrangement such as translocation or inversion, as I have already told. So it assists in selection of the embryos with normal chromosomal structure for implantation. But what is the current status of the assessment of the embryonic euploidy level? So when you're considering the PGTA, current good practice recommendations said no. They said pre-implantation genetic testing for aneuploidy currently says not recommended for routine clinical use for PGTA. But there is no other way around for embryo biopsy, and then later on for these PGT-M and PGT-R. You will have to perform embryo biopsy for finding out the abnormality level for mutation and structural arrangement. So this is the latest recommendation from the ASHRAE. We are waiting for the ASRM, what they are saying, and the Cochrane also. But later on, this Cochrane will summarize everything. But this is the current status. It is not only for the invasive, when we biopsy the tophotom cells and find out the abnormality level, but it is also these same guidelines also not recommending the non-invasive PGTA, okay, for routine clinical use. They says that these non-invasive PGTA is also under the research. They are no paper, there is no evidence till now that they are saying that this is the efficacy of the non-invasive approach is also not recommended by the latest guidelines. When we are talking about the DNA load from the mitochondria, they are also saying that mitochondrial DNA load measurement is also currently not recommended for the routine clinical use. Okay, so what is the future direction and conclusion? So future direction says PGT holds promising advancement, including the development, testing methods, enhanced embryo selection algorithms, and personalized genomic medicine, which will further revolutionize ART and improve patient outcome. But effective patient counseling is crucial to ensure informed decision-making regarding PGT. It involves discussion, the purpose, risk, benefit, limitations, and the genetic testing, as well as the exploring the alternative options and addressing the patient concern. Understanding the concepts of the ploidy and the genome is essential for comprehend the overall need of the pre-implantation genetic testing in ART. Specified PGTA like PGT-M and SR may offer numerous advantages in selecting genetic normal embryos, improving IVF success rate, and ensuring the birth of healthy offspring, but not the PGTA and NIPG. Okay, so these are under the research. Later on, we will have to understand that it is not only that whenever we are talking about this PGT testing and PGT screening and everything, what is happening at the level of the overall practice and the recommendation? Where are we exactly? We need to understand this. Thank you very much.
Thank you, Dr. Yogesh. It was an excellent excellent talk with precisely to the point and very well explained.
Any question from anyone, madam, if they want to ask, they can ask.
I think we'll take the questions afterwards, Dr. Yogesh,
No
because
problem.
we have like the other speaker online now,
Yes.
Dr. Meenu.
Thank you very much. Thank you, especially Talwar sir. Thank you so much.